Are you there now? Can you see it?
No, we're there, but we can't see it.
Oh, sorry, I don't know why it's...
It was there before.
Yes, it's just not choosing to...
Yes.
Oh.
Okay, all right.
Yeah, leave it.
That would be fine. Okay, go.
Good. I ask you to picture the scene of a group of thoroughbred mares, heavily pregnant and possibly carrying the next Melbourne Cup winner, such that the individual value of the foal is potentially worth millions of dollars. These mares are grazing in paddocks surrounded by native bird life, sharing the space. However, what if the space is the point of transmission of a bacterium that causes the mare to abort her foal? And what if the workers handling the affected fetal membranes become ill too? Recently, an outbreak of bird chlamydia was responsible for 1 in 5 foal deaths, and several people also became ill. Why is bird chlamydia, with a known preference for the respiratory tract, now showing a definite preference for the horse and the reproductive system, with late-term abortion being the only clinical sign? This forms the basis of my PhD as I seek to understand the infection dynamics of Chlamydia psittaci in the thoroughbred horse. I currently lead a 2-year longitudinal study which follows a group of thoroughbred mares, testing them and their newborn foals for chlamydia. I have detected for the first time chlamydia in healthy newborn foals at a prevalence of 14.5%. The chlamydia passed through the pregnant mare undetected, infecting the unborn foal. This suggests that the mare is not to be a good indicator of fetal chlamydial infection, and this information can be used to improve diagnostics. I was also sampling birds from the same paddocks to confirm if they are the source of chlamydia. Using DNA analysis, I observed that strains causing disease in horses, parrots and humans are identical, whereas a strain I detected in an Australian white ibis was different to the abortion cases. This may mean that some strains cause abortion. Detailed molecular work is required to further investigate this. I then asked if there were potential risk factors for the mares and chlamydia. Through multiple logistic regression modelling, I determined that the time of the year the foal was born was critical to chlamydia infection. However, it was not only the foals, it was the birds as well. All positive results occurred in the winter months of June, July and August. Identifying a time point of 3 months, considering that a mare is pregnant for 11 to 12 months, can focus intervention protocols. Discovering the drivers and level of infection required to cause disease is the focus of the second year of my PhD. The fact that healthy newborn foals have tested positive to chlamydia has created a new question. What is the impact on the young foal, this future elite athlete born with a respiratory pathogen? I hope to explore this new finding more. I feel very privileged to being invited onto these farms and help an Australian industry that's worth $9 billion to our economy and employs 70,000 full-time jobs. Furthermore, this work is designed to provide answers that will potentially prevent loss of life and minimise a zoonosis to people. Thank you.
Perfect. Thank you very much. Okay. Can you remove your screen, please? And Uma, can you put on your slide?
Yeah, sure, Abigail. Is it visible?
Not yet. Did you press
Will
share? Yes, now it's visible. Perfect. Go.
Okay. The title of my thesis is to understand the pattern of deleterious mutation load in human population. And in this, in order to understand my thesis, we have to look at two basic concepts, which is the genetic admixture. In this genetic admixture, we could say that there are two distinctly isolated populations. At some point, when those two populations interbred with one another, it forms a new population, which is called the genetic admixture. And the second concept is the deleterious mutation, which is, this is something which affects the fitness of a living organism. For example, the disease-causing mutations. And in every human possesses at least hundreds of deleterious mutation in their human genome. And based on the individuals, the mutation load differs. Mutation load is nothing but the accumulation of deleterious mutations. And based on this, we have formulated the aim, which is how this admixture influences the deleterious mutation load in a human population. So the objective is, based on this, we have formulated like quite a few, 3 objectives. So in this, the first objective is, for example, the parental population. One of the parental population is having a high number of deleterious mutation load, whereas the other one is having the less number. So in this case, we could expect the offspring will have an intermediate one. So based on this, like we'll be comparing the ancestral population's deleterious mutation load as well as the admixed population's mutation load. So that's going to be my objective 1. And the second objective, in the second objective, we have 2 scenarios. So the first one is, we have, in one of the parents, we have, 4 heterozygous deleterious mutation, because in this, we are going to be dig deeper into it, and in the types of deleterious mutation. So in this, like one of the parent is having 4 mutation and the other one is having 2. So in this case, the offspring will have 1 chromosome from each parent. So it is expected to be intermediate again. So by this, we could say that the mutation load will be reduced because of the admixture. And the second scenario of the second objective is the homozygous deleterious mutation, in which both the parents have the homozygous deleterious mutation in different positions. The offspring is subjected to have get this heterozygous deleterious mutation. So we could say that the homozygosity is getting reduced when 2 different populations intermixed. And the objective 3 is going to be the continuation of 2, of course. And in the admixed offspring, first time in the first generation, it will have 50% of chromosome from father and mother. And then after few generations, when the admixed individual has been interbred with the same population, so the remaining parental population's level will be diluted. So that's what it is. Yeah. I'm happy to take if there is any questions.
Thank you. No, there's no questions here.
Okay, all right.
Yeah.
Yeah.
Thank you very much.
Yeah.
Okay.
I'll just remove the...
Yes, can you both? Thank you. And Soheila?
Yes.
Can you put your slide on, please?
Sure.
Okay.
Could you see?
Yes. Go.
Okay.
Go.
Hi everyone, my name is Soheila Beiranvand. First of all, I would like to thank the GeneCology Research Center for providing this opportunity for me to come and present some parts of my work. I am in the early 3rd year of my PhD with the title of the Modulatory Effect of Australian Honeys on Inflammation, Oxidative Stress, and Wound Healing. Chronic wounds are defined as a wound which hasn't healed in the range of 4 weeks to more than 3 months, and represent as a silent epidemic that affects around 400,000 Australians at any one time. Approximately 2% of Australian national healthcare expenditure is spent annually for treating wound complications. Honey as a bioactive product comprises of mostly water and sugar, but also contain minor constituents such as phenolic acid and flavonoid, which are contributed to its antioxidant activity. Antioxidants are postulated to help control wound oxidative stress, and thereby accelerate wound healing. The wound healing process entails complex interaction between different cell types such as macrophages, fibroblasts, and keratinocytes. However, some studies investigate the effect of honey on wound healing process. They mostly focus on its microbial activity. To the best of our knowledge, there is only one study that determined the effects of only one Australian honey on pro and anti-inflammatory cytokines. However, this study used a monoculture system, which doesn't permit investigation complex interaction which occur in vivo system. So to resolve this matter, we decided to use a quasi vivo system, which is a unique system, to study the interaction between different cells which are implicated in wound healing. As you can see, the system comprises of different chambers and in which different cell types can be grown. Also, the chambers are connected to each other by tubing, and the peristaltic pumps enable continuous perfusion of the chambers with culture media. This system allows sequential movement from one cell type to another with the culture media and also cellular secretion. It's hoped this study will lead novel therapies for managing patients with chronic wounds. Thank you.
Thank you very, very much. If you can remove your slide, please.
Sure.
Mikaela is next.
Yeah, can you guys see that okay?
Yes, go.
Awesome. Hi everyone, today I'm going to be presenting an overview of my honours project investigating important factors in golden orb spider silk. Spider silk has always been valued due to its combination of mechanical properties, such as toughness and flexibility, outperforming both natural and synthetic fibres. This unique material is also lightweight, biodegradable and biocompatible, and has potential medical uses as it doesn't produce an immune response when implanted into patients. As such, it has long been the goal to produce it on a commercially sustainable level. However, creating a synthetic silk able to match the impressive mechanical properties of natural silk still eludes researchers, but not without lack of trying. Many different methods have been tried, including transgenic goats, which you may have heard of. That company did end up going bankrupt. The main component of spider silk are these large proteins called spideroins. There has been a heavy emphasis in literature investigating the structural and functional properties of these spideroins. However, there are many small proteins present in silk that have not been widely studied. For my project, I'll be trying to investigate important factors in spider silk that may have been overlooked in previous research. The spiders I am investigating are a species of golden orb weavers called Trichonephila plumipes, which I've got a picture of on this slide here. A large one is about a handspan in size. They are commonly found in Queensland bushland regions, including the Sunshine Coast, and you might have a few hanging around in your backyard if you live near the bush. They're called golden orbs due to their silk having a distinctive golden shine. Anyone who's accidentally wandered into their large webs would have discovered it's a lot stronger than it looks, which I've done before, which is why golden orb silk specifically has been the focus of much research. These spiders produce different silks for different purposes. I'm looking at dragline silk produced by the major ampullate gland, colloquially known as the silk spiders use to hang menacingly from the ceiling. It's the toughest type of silk, being as thick and resistant as steel, but is more flexible. I'll be identifying small proteins that are likely to play a role in the synthesis and structure of silk. To do this, I'll be analysing silk samples collected over a period of several weeks and finding those small proteins that are consistently present in all the samples. I'll also be comparing the silk between spiders carrying an egg sac and those not, which will eliminate those small proteins that are the result of a reproductive cycle rather than being integral for silk. A holistic understanding of all the components of silk is required to be able to reproduce it to the same standard as spiders. If spideroins were the only component required, spider silk would be relatively easy to synthesize, which is not the case. Establishing the integral small proteins in silk will allow us to produce a new hypothesis for silk synthesis and help to untangle the complicated web of spider silk structure. Thanks.
Thank you very much. Next is Salma. If you can unmute yourself and put your slide up.
Can you see my slide?
Yes, we can. Okay, go.
Inside each of us right now, cancer-like cells are growing. Luckily, this is not as bad as it sounds because most of us have healthy, active immune system. When one of our cells becomes damaged or undergoes mutation, our immune system wants to take the cell out and develop a mechanism to neutralize or kill the cell before it grows into clinical disease. This mechanism is coded in a chemical called DNA. A particular segment of the DNA, what we call gene, play the key role of our immune system. In this slide, the highlighted section is a BRCA gene, which control breast cancer. Unfortunately, this mechanism is a foolproof system because tumor cells are clever. Over the time, they can change their appearance by adopting disguises through a little modification of the genes and allow them to evade immune control. However, due to rapid technological advancement, a large number of cancer-related genes and their sequences have been identified, and most of them remain unexploited. However, the important driver genes, which employ a discriminatory advantage to cancer cells, are usually very small in number. Therefore, an approach of prioritizing modified driver genes may help to understand the disease development, to take further treatment measures. In my PhD, I aim to develop a holistic management strategy for 4 women cancers: breast, ovarian, endometrial, and cervical. I identified the key driver genes controlling all these 4 diseases. I used multiple bioinformatics softwares that enable the analysis of this large number of data to identify genetic variations associated with cancer genomic studies. For the first time, I identified the common genetic mechanism between these 4 women cancers through functional analysis of the genes. I interpreted their role on disease development. I also conducted an expression analysis of the identified genes using data on disease appearance around 5,000 patients around the world. My results provided a list of evidence-based genes in each of these cancers. My finding will likely lead to the development of effective prevention, diagnosis, and treatment strategies for women-specific cancers. We believe that this approach could lead me to the day when I will be able to say to all of you, we all have cancer-like cells, but none of us have cancer. Thank you.
Thank you very much. I certainly hope you get to that day very quickly. If you can remove your slide, please. And the next is Ryan. Thank you.
Hey guys, can you hear me pretty well?
Hold on and go.
Hi guys, my name's Ryan. My honours year began with a pandemic. I mean, what a way to further your education and really start your academic life, right? So I read the papers and the news articles day in, day out. There appeared to be this common line of thinking. It was that the more people were tested, the better chances of restricting further spread. So they tested, and then they ran out of tests. And then the news stories told us that the tests weren't really even that good anyway. In a global pandemic that affects people from all different demographics and socioeconomic regions, access to cheap, fast, and accurate tests could really mean the difference between small-scale spread and global devastation. Currently, one of the world's small-scale spread issues is Zika virus. Now, this nasty little bug currently has no medicine to treat it, nor does it have a vaccine to prevent it. It's also believed to cause what's called Zika babies. This is devastating newborn birth defects such as underdeveloped brain and cranial features, as well as central nerve system malformations. So why should we worry about testing for something that we can't treat? Well, this virus is initially transmitted by mosquitoes, and once it's inside the host, it can be passed on sexually and, worse still, from mother to unborn child. So in theory, a testing regime could not only provide valuable information to entomologists, they're the bug nerds if you didn't know, but also influence decision-making processes concerning conception, potentially reducing those numbers of Zika babies. All right, great. Get some tests out there and get it done. Now, this idea is much easier said than done. Traditionally, tests, cheap does not necessarily align with accuracy. Now, current methods of diagnosis are ultra expensive, they're time consuming, and they often require really highly skilled people to carry them out. The aims of my research is to develop a simple, rapid point-of-care test that provides accuracy needed for diagnosis, coupled with the low cost to ensure affordability for everyone. With the help of the Australian Infectious Disease Research Centre at UQ, we're currently utilising genetically modified viruses that are not dangerous to humans to produce a dipstick-style test that will identify and indicate the presence of antibodies in a patient's blood, confirming if and when they've been exposed to the virus. With increasing interest currently being shown by international investors, the development of a test is underway and it's showing really positive results. Further work will ensure that production of a test could rapidly reduce global numbers of devastating outcome Zika virus claims annually. In a time of global division, we really need to come together, focus, and assist those who need it most. My research, it has the potential to provide a solid platform on which to bounce from the next time the common line of thinking is the more people are tested, the better chances of restricting further spread. Thanks.
Thank you very much. If you can remove your slide, and Kate, if you can put up yours, please.
That's okay, is that working?
Yep, fantastic, go.
Okay. You may not know it, but Australia has been besieged by invaders. These invaders don't have any arms or legs, and their main weapon is slime. They're hitting us where it hurts, in our food basket. So in southern Australia, where we grow important grain and legume crops like barley, wheat and oats, 4 species of European snails survive the intense heat of the summer by climbing and excavating or sleeping on the crops, contaminating the harvest, causing economic losses and severely damaging Australia's reputation as a quality grain supplier. So the best way to fight any invader is by learning their secrets, but to do this we need to work out how they communicate, maybe by following the trail of slime that they leave behind to find some clues. So what's in snail mucus that we don't know about? We know that it's 90 to 95% water. Could snails be communicating through it? We do know that many species of snails will follow the mucus trails of conspecifics. We know that other molluscs use proteins as pheromones to aggregate, to mate, or to aggregate and mate. The research that I've done over the last 16 months using sophisticated molecular methods, such as liquid chromatography, mass spectrometry, shows that yes, there are proteins in the mucus. Besides some structural elements like collagen and actin, a few other proteins where the function is unclear. Unfortunately, like any good invader, these snails don't give up their secrets easily, and as yet I haven't been able to identify a protein pheromone. There's also a possibility that these snails might use a volatile pheromone in the same way that insects and mammals do. In other words, using the smell of the mucus to communicate. And yes, my research using gas chromatography mass spectrometry shows that there's a number of volatile smells coming off the mucus trail, like undecane and sulfatone. And these have been known to function in communication in other species as diverse as rats and aphids. So I've not yet identified a volatile pheromone because this involves the dreaded behavioural assays. And if we know one thing about snails, it's that they are slow. But I'll persevere because if I can find a snail pheromone, this might be just the weapon that we need to control pest snails, in a similar way to how pheromone control is used for insect pests. So my results to date are telling me one important thing. The mystery of snail communication and the role of the mucus trail can and will be unravelled. And this could lead to the development of an effective, sustainable biocontrol method and the ultimate protection of our important food crops and our reputation as a grain supplier. Thank you.
Thank you, Kate, very much. Okay, next, T. Are you ready? Kate, you need to remove your slide.
Got it.
T, are you ready?
Yeah.
Okay. Put your slide on, please. Okay, go.
Yeah.
Yes.
Good afternoon, everyone. As you know, white spot disease in prawn is a name I suppose many of you have heard before. This disease is widespread through farming regions around the world and recently caused a severe loss for prawn production in Queensland 3 years ago. Currently, there are several treatment methods being applied. However, they have all failed to fight against the disease. So, what is a sustainable method to improve productivity even when the disease occurs? This is the question that my thesis looks for the answer. Based on the advance of genetic breeding selection, we attempted to establish a new population of prawn which resists to the disease to improve the survival in farming. At first, we analyzed genetic variation of white spot disease resistant trait to find out if this trait is heritable in different stages of development. Our findings show that there is a moderate inheritability at early development phase for this trait. From the first result, we carried on breeding selection experiments for improved resistance for this disease. After 2 generations of selection, we found that there is a 12% of higher survival in selected families comparing with unselected ones. On the other hand, recent developments in high-throughput next-generation sequencing platforms have opened new possibilities for improving disease traits based on DNA information. In our study, we focus on genotyping to identify potential molecular markers that are associated with white spot disease resistant trait, and then applying for breeding selection for high WSHV resistant in white leg prawn. So our findings are preliminary for application in disease-resistant selection. Therefore, it will produce high-quality seeds for prawn farming. It will maximize output for farmers, bring happiness for so many lives. And for someone who loves prawn like me, don't need to care about the price, always enjoy the succulent prawn dish on the table whenever we want. Thank you very much, everyone.
Thank you, T. Yep, prawns are amazing. Okay, can you remove your slide, please? And Lockie, you are last but not least. If you can put your slide up, please.
Yeah, it's that one.
We can't see.
Is that the right screen?
Yes, that's ready. Okay, go.
Second, sorry. Okay, go. Hello, I'm Lachlan Dennis, and I'll be talking about using DNA barcoding to identify larval grouper diet. Grouper are important aquaculture species. Most farmers source small fish from the wild between 2 and 5 centimetres called fingerlings. They're captured and grown into adult fish. Most fingerlings don't survive, and many grouper species are endangered. Full cycle aquaculture is feasible for grouper, but the larval stage before fingerlings has proven difficult for farmers. Larval grouper are less than 2.4 millimetres when they hatch, and their mouth gape is less than 200 microns. They also only eat live food, specifically marine zooplankton. Survival in farms is very low for the larval stage, often below 1%. Little specific information exists on larval diet. The prey is microscopic and often too digested to identify. Molecular methods are possibly the only way to determine specific prey species. DNA barcoding is the ideal technique. It works by identifying a single gene sequence from a sample and then comparing this gene to a known database of sequences. In mid-2017, I travelled to Vietnam and collected larval grouper from our collaborators at Rio 1. These larvae were reared in open water ponds, seeded with zooplankton, and had their choice of prey. I collected larvae as well as water samples daily so I could compare what larvae could eat with what was actually in their bellies. In Australia, we used degenerate primers to target a 365 base pair fragment of the COX-1 gene. We tested these primers on a range of zooplankton, optimized the protocol, and performed Sanger sequencing. We hit an obstacle. We couldn't determine the stomach contents because the larval DNA was so abundant, all sequences were identified as grouper. So we decided to use next-gen sequencing. I modified my original primers with Illumina MySeq adapters and had the sample sequenced. For each time point, I got 300,000 reads. The majority of these were grouper. This would take about 500 hours to blast just for a single time point. I improved upon this using a bioinformatics pathway design for metagenomics, from the initial 300,000 reads to a final cluster of 2,000 reads. Each cluster represents an operational taxonomic unit with sequences 97% similar to each other. Each read also has a count indicating how many sequences it represents. Blasting these sequences takes about 2 to 3 hours per sample. The blast result shows which species are present, and the count shows the abundance of each species. At different time points, I can see how this changes over time. This is the step I'm up to now, running blast on my samples and working out the relative composition of each. In the near future, hopefully, I'll be able to provide the first clear picture of which zooplankton are important for larval grouper and enable farmers to transition to full cycle aquaculture. Thank you.